Hi everyone, thank you so much for joining our ChIP webinar today. My name is Rebecca Northeast and I am the Product Manager for Protein Tech. With us today we have Dr. Poolman, who is going to be leading the workshop, but just quickly going to start with a very brief company introduction. Okay, right, so just want to briefly introduce the Protein Tech group. So Protein Tech is the main company of Protein Tech Group, and we were founded in 2002 by Dr. Lee, and we primarily sell antibodies and ELISA kits, and all our antibodies are manufactured in-house and that undergo extensive QC. We also have around 4,000 antibodies for immunoprecipitation and over 200 antibodies which have been published by customers in ChIP. As well as Protein Tech, we acquired the company Humankind, and they were founded in 2005. And Humankind sells human cell-expressed recombinant proteins, primarily for stem cell and gene therapy. And the reason that Humankind sells Humankind proteins are excellent is because they are produced by the HEC293 cells. Therefore, they have native folding, maturation, and glycosylation, meaning that they have much better activity. We also have Chromatin, and we acquired Chromatin in 2020. And Chromatin are a nanobody company, primarily making nanobody reagents for immunoprecipitation of tagged proteins. And they sell the flagship product called GFP Trap, which has been used in many different immunoprecipitation experiments, including over 60 publications in ChIP. And also just wanted to announce our partnership with Active Motif. And so Protein Tech is actively collaborating with Active Motif, which is a well-known epigenetics research company. And therefore, between us, we allow customer solutions. We are offering customer solutions for their entire ChIP workflow. And on the call today is Mike, a product manager from Active Motif. And at the end of the webinar, Mike, myself, and Astrid, who is the product manager for Chromatin, will be able to ask any questions you might have about our products. So now I just want to introduce the speaker, so Dr. Poolman. So Toryn started out as a postdoc with Professor David Ray for 10 years, between 2011 and 2020, starting at the University of Manchester before moving with David to the University of Oxford. Toryn was researching on glucocorticoids and circadian biology, and during this time he did a lot of ChIP experiments. Toryn is now a teaching fellow at the University of College London, and he also has an interest in complex proteomics data sets, as well as creating reproducible workflows in R. So that's it for me. And now just to hand over to Toryn and get you started on the ChIP workshop.
Hey, thank you very much. Thanks for joining. So today we're going to go over some of the aspects of a ChIP workflow. And this is very much from the point of a beginner. So a lot of the things I'm going to talk about are going to be more at the kind of really at the beginner level. Just to kind of take you through each part of the workflow, some of the things I've discovered on the way. So we're going to go through sample preparation, the immunoprecipitation part, how we quantify this stuff at the end, and a tiny bit about ChIP-seq. And then I'm going to hand over for some information about antibodies. Oh, whoops. Hang on a second, just stopped on the, so now we're going to get, let's go back here again. Apologies. And then we're going to hand over some information about nanobodies for ChIP as well. And then we'll have sort of a Q&A session at the end where you can ask as many questions as you like. Okay, so this is kind of beginning bit, a little bit introduction about what is ChIP. So this is obviously a highly simplified diagram. But ChIP is basically chromatin immunoprecipitation, is looking at protein DNA interactions where we fix protein and DNA together as a complex, and then we kind of purify that complex with an antibody. Then we remove all the protein, and then we measure the quantity of DNA, and we're looking for specific sequences that the proteins are binding to. Now, if you were to do the same thing, so fix your DNA to your proteins, and then get rid of the DNA and measure the amount of protein there, either by mass spec or like Western blot. That technique is sometimes called RIME. So It stands for rapid immunoprecipitation of endogenous proteins. And that's kind of a sister technique, shall we say, but essentially they're both looking at the same thing. Just one removes the protein and the other one removes the DNA. And that's basically all ChIP is, in a nutshell, is looking at DNA-protein interactions. So actually, only to a few days ago, I actually thought to myself, so what were the original ChIP papers and what did they look like? And I was quite surprised actually to see how few citations, the papers I could find that were first publishing the idea of looking at a protein DNA interaction and then a sequence in this case. And they were published in 1988, one of them, and it then directly cites another paper from 1986 about the kind of immunofractionation of chromatin. And I was actually also quite surprised how complicated some of the papers were. And I didn't really, it kind of occurred to me that I didn't know as much as I thought I did about purification of histones either, and what a kind of complicated world that was. And this is kind of, to quote a phrase that I've stolen from someone else a little while ago who teaches jiu-jitsu, science can always make you feel like a beginner. And no matter what the technique is and what you're doing, it's very easy to suddenly feel like you are a beginner really, really easily, because some of these things are extremely technical and they can be long-winded. And it's quite easy just to have that kind of serious imposter syndrome. No matter how long you've done this for, it can easily make you feel like you're back at day one again. So if you're like me, look at a lot of papers and often feel a little bit overwhelmed by what I'm seeing complicated graphs, lots of Venn diagrams, heat maps, various things like that. And it almost feels as if the people who were doing these techniques were some kind of wizards. They were these kind of super beings of science. And, they create these amazing publications in super high impact journals, and I thought, I'm never going to get there. And I guess really, you're trying to always break down the kind of voodoo of science in some respects. You're trying to sort of understand that it is not this kind of super beings doing science who get these results. It's just a question of how you can break this down into its simplest forms to then master each part of a technique. So a lot of techniques we do in the lab these days, albeit ChIP or proteomics or sequencing or even Western blotting still, you really have to know the phases of the technique and really kind of try to understand from an empirical level what you're doing at each part of the technique. So if you want to be successful in a technique, I always think it's really important that you really dive in to the mechanics of it. And, it's the idea of practice as well. So to move on to my second slightly woolly point, shall we say, is that, a new technique can be a challenge and you really have to practice these things to really get a feel for it. Now, analogy I quite like is just because you go and buy a surfboard and you're in front of the sea, doesn't mean to say you can surf. Because you go and buy a kit, doesn't mean to say that you can apply it straight away and you'll get it to work either. Now, I've tried surfing, I can tell you, it didn't go very well. But this is kind of my point. You really have to spend time working on a technique, really trying to understand it, add practice and elements of practice into it, like you would anything else. Like if you were trying to learn to surf, if you have that sort of analogy, I think that's how you actually get really good at something. Now, I may be teaching the psych thing, you may understand this already, but I think it's taken me a long time to go work that out myself. That really is about the idea of practicing something is really important. Okay, now I've lectured you all of you about this. So where to start? So if you're going to start ChIP, just say you want to do some ChIP, you've got some cells, you've got bits of tissue. It's really important that you start off with something that's readily available. So you don't go and get your most precious samples and start trying to chip away if you've not done it before. Of course, you always start with something that is readily available. Say a good cell line, for example, that you can get limitless amounts of cell, or if you've got lots and lots of spare tissue, I would always start off with that, just for the first instance. Now, you don't want to make that your project. Your supervisor will not be happy if you spend 6 months on the practice material and you feel like you're getting nowhere. That's kind of, that is, I think it happened before. I think it's much better to have a few good practices and then go back into your what would be the tissue that you're actually going to start with. And this may seem kind of obvious, but actually, I think at times when we're working in the lab, it's easy for us to get a bit ahead of ourselves, and we need to step back a bit and just go for something that we actually know may work. So if you're going to start ChIP off relevant material, you start off with something like an antibody that has actually been shown to work. So a really good one is the kind of H3K287 cetylation antibody, something like that. And you start off with primer sets that you actually know also work as well, so you buy them commercially in. Now, ChIP is a long technique, in relative terms, for molecular biology, I think, anyway. It takes about a week to get to the end. And I'm sad to say that I do have a freezer full of unfinished ChIP experiments that only got to a certain stage, ones that I froze at one stage at the pellets, ones that I sonicated, ones that I got so far with. And it is easy that to happen because as lab scientists and as postdocs or PhD students, we have a lot to do, a lot to get through. Sometimes things crop up, sometimes you have to do presentations or whatever. There'll be other projects that come along and things will get stuck behind. And when it's a long process and it takes a week to do something, it's easy to have a graveyard in your freezer. Luckily, this isn't actually my freezer, but I've got this image, but it would look a bit like this, full of boxes of things that were chipped and not been chipped. Okay, so here is the workflow. Okay, so as you can see, it's quite long. Now I've split this up into 3 acts. Okay, so Act 1, sample preparation. Act 2, the immunoprecipitation. Act 3, the quantification stage, however you do that. And I like these kind of 3 segments, mainly because I feel as if these are quite good pause points. You can pause ChIP at various points, actually. Admittedly, you could just pause it straight after you fix your tissues or cells. But I always like the idea of going straight through to the sonication. So fixed lyse sonication. This sample preparation stage, take your cells, fix them with a suitable fixative, lyse them, and then you isolate the chromatin after the lysis, and then you break up the chromatin up into the fragments using sonication. The Act 2 part is the immunoprecipitation part of things. Here, so we're going to capture out the complex of protein and DNA. So we add the antibody, capture the target. Then we have a secondary purification, which is the antibody itself. So we use a suitable bead-based system with a kind of protein A or protein G to capture out the antibody and protein complex. Now, immunoprecipitation itself is a separate technique. We're looking at co-binding proteins, and that in itself has, can be fairly involved and can be quite difficult. Then we go on to the 3rd stage of things, which is the quantification stage. So we want to quantify how much DNA we have, especially if we're doing ChIP sequencing. And then we move on to doing some sort of testing, whether we've actually got the sequence of interest using qPCR maybe, or even digital PCR, which I'll tell you about in a moment. And then we can move on to sequencing. So these 3 lots of 3 are fairly involved. If you think about it, the sample preparation stage, that's just maybe like a nuclear prep. And then we have an entire immunoprecipitation stage, and then we have an entire lot of quantifications to do. So this is a lot of stuff to get through. So sample preparation. The first step is to fix the tissue or cells. And primarily this is done with formaldehyde. That is the most commonly used fixative for ChIP. Usually about 1%, and usually we fix also between, say, 5 to 10 minutes. If it's cells, it's quite simple. You just literally have to add the formaldehyde to the cells. If you have tissues, you actually need to chop up the tissue into fairly small chunks and allow the formaldehyde then to get into the tissue itself. Now, there are other fixatives that are available, which people do use, and there's also a trend of using 2 fixatives, so you can use a double fixing stage. Usually you would then fix with one of these other fixatives. So these 2 examples I've got here, one is called DSS and the other one's called DSG. These are really commonly used fixatives in mass spec, and now there's actually a fair few studies I've seen where they do a double fix. So you fix with DSG, then with formaldehyde, and then carry on the process. And it's been shown to improve the fixing for some targets. The difference between these fixatives are some of them have longer spacer lengths. So formaldehyde is a fairly short space length, and the spacer lengths and these other 2 compounds are a lot a lot larger. Now, I think when you're starting off a technique like this, I would just go for one fix, unless you have a specific reason, or you found a paper maybe, and you're quite confident that you need to use 2 fixing steps. I've seen this for P65. I think anyone works on that before. But first of all, I would start off with literally just one fix. And if you're going to start off with cells, make sure you have enough cells. So don't go trying to do lots of chips at a small scale level at an early stage on. Your first aim with the chip experiment is to get something to the end and to get some sort of result that showed you could do the technique. So I would always go with a lot more cells, say 10 million plus cells at the start, or a nice sized bit of tissue. And then I'll make sure I have enough samples to cover any disasters, so at least 6. I wouldn't go for like 60 samples because that would mean a heck of a lot of work, and you put yourself in a lot of pressure. You have to go for a reasonable amount of samples you can get to the end to. The main point of this is to get to the end. So you just need to have, say, 6 samples, enough just to test, and start off with formaldehyde and go from there. Now, you can, of course, buy kits, some of these things, which will include a lot of these reagents, but you don't have to buy a kit at first. I appreciate that sometimes lab budgets don't always fully extend to every eventuality. And there are lots of published methods that I'll come on to in a second, but it's more important at this stage just try and get something finished. So single fix is preferable at first, I think, just for a test session. Be aware that there are other fixatives available that you can add in, and it may help some things that are maybe more difficult to chip. Okay, so the second stage is to isolate some kind of chromatin. Okay, so you may have done before a nuclear prep, where you were just trying to see, for example, maybe you're looking for nuclear translocation. Maybe you're looking to see whether a protein ended up in the nucleus. And if you had a go at that before, you may have discovered actually it wasn't massively easy at times, and it can be quite variable for lots of reasons. But bear in mind that the first part of ChIP is a bit like a nuclear prep in many ways, because it's fixed. Because you fix your cells, and you've obviously then cross-linked your protein in the nucleus, there's less chance of things kind of just washing out or falling off. But because it is fixed, it then becomes a lot harder to lyse. So the first steps of ChIP seem fairly kind of robust, and we have an initial lysis step, usually with a more milder detergent like Triton. Then we go through a sequential spinning, where we basically keep retaining the pellet, which where our nuclear material is. There's often a nuclear wash in some methods there is, and then we finally put it into a sonication buffer. Now, before that stage, we generally use a homogenizer or a tissue rupture to really get things broken up. So you need to make sure that you lyse the cells, and you can check this sort of lysis to see whether you've actually got nuclei, see that you've actually lysed your nuclei as well. That can be checked. You can, of course, do a kind of Western blot on your chromatin preparation to look to see whether your protein is actually in that compartment. It generally doesn't look very pretty. So when you do up here, I've got one of my old nuclear preps up here. This is the GR protein. You can see I've kind of isolated out the histone. That was, no great shakes there to isolate histone. But the GR protein here fluctuates around the cell in response to steroids. But that is actually, looks like a nice Western blot. I didn't have one to hand where I actually tried to Western blot out a kind of sonicated sample, or even a nuclear preparation once it's been fixed. It generally doesn't look as pretty as this. Again, it can look quite ropey, and it can be a lot of background, and your previously nice looking Western blots then don't look quite so good. But it is very important that you do, this is obviously a critical step, okay? You're going to isolate a nuclear contraction of the cells. Got to make sure you get good lysis. So definitely a homogenizer is really important. Some kits don't have the 3 steps. They will just have maybe more than 3 steps. They may have a wash step with PBS and a detergent, perhaps maybe, and then go through chromatin isolation that way. So if you are starting off with something like a macrophage or something like that, something similar, they can be very hard to isolate chromatin from, and you don't get very much. And some cell lines are also quite a challenge to get chromatin out of as well. This is maybe not biology, this is something else. Okay, this could just be resistance to detergents, and some other factors. That's why it's quite good to start off with a cell line that some people have actually chipped before. If you're doing something that's quite new and different, and you want to see whether you can get the process to work, I would start with something like a HeLa or a hex cell line, and just see how you can get things to kind of fractionate. And especially if you've never done a nuclear prep before, it's worthwhile having a go and just seeing what you get at the end. There are lots of good published methods for these first few stages. There's a good one here, methods. It's quite old now, but it does detail out each step very well. And it's worth spending some time looking at some more detailed methods just to make sure that you can get the nuclear isolation to work. So start with published method, get a kit, be wary that some cell types are difficult to sonicate and lyse, okay? There we go. I feel like I'm kind of instructing you all in some way, but this is my tips for kind of slightly more a better success. So once we've got our chromatin prep that we're happy with, we then have to sonicate it. So I'm not going to cover enzymatic digestion here actually of chromatin. Going to go straight for sonication. Okay, so the more common technique, I think. And the third decision you have when you want to sonicate your chromatin is whether to use a water bath or a probe sonicator. So you may have a sonicator in the lab already. You may not have a choice. You know, you're not going to necessarily have the chance to buy a brand new sonicator just for your own purpose. You may be lucky, you may be able to do that, but quite often you don't. So you have to go what you already have. I've used both water bath and probe sonication in the past. I favor probe sonication these days, but I'm not saying that water bath sonicators don't do the job, and I have got results off them in the past. But my go-to at the moment is always a probe sonicator. And if you're trying to break up your DNA into fragments, and you're trying to do that without destroying the sample. If you destroy a sample, you're trying not to ensure that it gets so hot during the process. So the process of sonication always leads to heat, okay? And you'll end up with a very hot sample. And it will always get hot, I'm pretty sure. I've used water bath sonicators with tons of ice and cooling systems before, and there's always some heat generated. You just have to basically do enough rounds of sonication resting on ice till you get to a phase where then you have a sonicated sample. Now, the sonication time depends on the tissue and on the cell type, and generally between 5 and 10 minutes at first is a good place to start off. You can do optimization experiments where you will then take out a small amount of your chromatin at each stage after several rounds, and then you will then analyze those on a DNA agarose gel to look at the size so you can roughly see. Now, these days, I actually favor a tape station to look at the sizing, but this is one from quite a long time ago, actually, when I first probably started doing ChIP, I think, to look at some of the sizing we have on the gel. Be warned that when you run DNA gels with DNA like this, that the salt concentration in there can alter where it appears on the gel. So you have to make sure you account for that. It can look really variable, as you can see here. Some of them, and look at my plots here, you can see that there's some that are quite low, around the 200 base pair mark, and some that have these streaks going up. And this can be quite common depending upon, obviously, this was a piece of liver tissue, chopped up into small amounts. Obviously, the chopping could have been different, the chunk size could have been different. There's lots of potential error I could have had in this. And it can look quite variable. And sometimes you just have a complete streak if you're not careful. And I was going to show you a really bad sonication, but I was a bit too embarrassed to put it in there because I think it was a day where I'd done some sonication, I got onto a gel imager, I couldn't see very much, and then I went to a UV box and it just looked terrible. So that's what so it was just not the best at all. If you have a particular sonication in your lab that is come on to that in a second, yeah, about that question in a moment. If you let me just go back one. Oh, I can't go back one moment. I can't go forward either. No, it's frozen. Oh, whoops. There we go. That's good. If you have a particular sonicator in your lab, so like maybe you're lucky, you've got an expensive covirus machine, you have to check the buffer conditions they're telling you to use. Just be aware of that. So the buffer conditions for sonication can be quite important. If you've got a specific machine that tells you to use a specific thing, then do stick to that. If you just have a pro-sonicator, you have a bit more freedom maybe, but just be aware that there may be some really important details in some of the kits you can have for sonication. So make sure you have the right one. I would always start the pro-sonicator because I think it's a little bit easier in some respects. Sonicate over a period of time, and then you'll be able to hopefully see something on a gel at the end. Okay, so once we've sonicated it and we've got a pattern on the gel, well, once sonicated, before we run it on gel, we need to know how much is there. So once we got our sonicated chromatin and we've removed all the particulates, then we need to purify some of that chromatin. So you take a small fraction of that chromatin and you call that your input. And you can purify that chromatin using sort of DNA column, like a PCR cleanup, or you can use solvents such as phenylchloroform, methanol. Be aware that there's some differences there between those two methods. For a column, obviously you get high purity, but you may get a lower yield, and some of the very small fragments may not be kept, just so you've completely obliterated your sample. You may not necessarily keep everything. I personally generally favor columns most of the time, unless I have tissue, where I would use phenylchloroform, where you do get high yield, but you get lower purity. So the other question here is, if you use a nanodrop to quantify your DNA, or do you use a fluorescent-based technique, such as like pico green or a qubit? Now, be aware that obviously we all use nanodrops every day, okay? But nanodrop versus DNA fluorescence techniques are quite different. One's just measuring DNA, one's measuring wavelength, where lots of things will absorb. You also be aware, I don't know if people realize this, but on a UV spec type nanodrop approach, that is actually quite insensitive to protein contamination. That method was originally used to detect DNA contamination in a protein solution, not the other way around. And you can have a lot of protein contamination without a great shift in some of the ratios. So be aware of that, of course, as well. If you have an extremely low concentration of input at this stage, and I've seen this happen a lot, okay? People go through this process, they take a bit of chromatin out, they purify that DNA, they go to the nanodrop, and it says they've got like 2 nanograms per mil, a microliter, something like that, basically, some very low quantity. Sometimes you haven't taken enough, you hadn't have enough cells there, and the fraction you took at the beginning wasn't enough. And that basically means that you won't get a very good reading on the nanodrop. So make sure you take enough input. If you haven't got very many cells there, do make sure you take enough at the start. So take 100 microliters, of course, because you need to know this. You have to know how much you have in the input. That's very, very important. Obviously, because you've got chromatin, you do add RNAs and then proteinase K, and make sure you only add it in that order. Don't kind of proteinase K it, forget about the RNAs, then go and do it later, because the RNAs, the quantity we add into these assays, have some very unusual binding patterns to DNA. So you have to make sure you've got RNAs and proteinase K, then purification, then quantification. And if you always do a qubit, then always do a qubit. And if you're always doing nanodrop, then always do a nanodrop. Don't start flicking around, because there are different results you're going to have at the end of this. Okay, so what was my points here again? So yeah, make sure you have enough input to quantify, and be warned that ChIP is very sensitive to different input quantities. So if I have one sample where I have 10% more than the other one, then it can make a difference at the end to your results, because we're then dealing with a very sensitive measure at the end. Okay, so just bear that in mind. Okay, so now on to what used to be the bane of my life, the immunoprecipitation, which is a bit of a challenging technique, to be fair. So antibody selection, just before we go on antibody selection, okay, so you're going to pick an antibody for your ChIP experiments. I would start off with something that was been readily used before. Now, if you have a target that is new maybe to you, you need to make sure that the antibody can actually be used in immunoprecipitation. It may not be known, so you have to try it out, just do a regular IP for that. But there's lots of information on antibodies these days, and this one particularly. I say this is my favorite antibody. I shouldn't really have a favorite antibody, but I do. Favorite antibody because it is basically suitable for all sorts of different techniques. It's been validated in a knockout as well. And it's been published about, I think there are 52 publications for this one. Okay, so loads of publications, validated in multiple ways, that's a good antibody to start with, okay? There isn't any, unfortunately, there's no guarantees, like anything in the lab. There's no thing 100%, but it gives you a very good chance, if you have an antibody that you know definitely works for immunoprecipitation, that it may work for ChIP, hopefully. And that is a good start to take at the beginning. Look for saying that's been well published, well used. You can add multiple antibodies into one ChIP. I'll come on to that in a second, but you can pair them up, but you have to make sure that that definitely works, okay? But if I was starting out, I'd almost certainly go for one antibody on something that has been published, and that will give me a good chance of getting a good result at the end. So in immunoprecipitation, so we take a quantity of chromatin, of course, we measure that as DNA, and then we incubate that in the sample. Now, I never do the trick where you pre-bind your antibody to your beads. I don't do that, okay? I always go for, take my chromatin, calculate the quantity in there, add the antibody, and leave that overnight. Usually that works out quite well in the protocol because you have an overnight step. And you're not trying to cram it all into one day. We need to have pause points. Very important. You've got to have pause points because otherwise we're trying to do a lot in a small amount of time. So with a immunoprecipitation, just say you're looking for a complex, a protein complex. So IPs can be, they can be difficult. So there's lots of factors. In an IP, it's a struggle between specific targets and non-specific targets, and you always get lots of non-specific targets. The non-specific stuff is binding to other things in the media. So, for example, we'll add in our beads to collect our antibody, and with that, you end up with lots of binding to that resin, or maybe the antibody picks up different things, or maybe the process of incubating this stuff for a long period of time then means you get denatured protein, and that forms a bridge to bind other proteins together, and we end up with this big mess of stuff. Unfortunately, that's what we're looking at. And it can be very variable, okay? So I have my antibody to my chromatin, I incubate that overnight, then I capture that antibody with a suitable bead resin, like I want to in a second. We also use a spiking control. So basically, immunoprecipitation is extremely variable. And to get around that, we've got to have some way of normalizing between samples. Each sample is dealt with very differently. It's not like an ELISA plate where you can just wash everything in one go, do checkerboards. It's not like that. It's nowhere near as simple as that. There is lots of potential for bead loss. There's lots of potential for things to go wrong in this process. So having something we can put in, i.e. the spiking control. So we put in, we buy from active motif cell this, they Drosophila chromatin that we put into the reaction at a fixed amount, and then we IP out that chromatin with a separate antibody, and that gives us a control for the immunoprecipitation. It is quite an important step, I feel, this, because it's hard to know whether your errors in the system are just kind of washing bead loss. It's very difficult to know at times. And I've done so many IPs over the years for mass spec, for chromatin immunoprecipitation, and it is quite a challenge at times to get consistency for lots of reasons. And having a spiking control is a really good idea. So I definitely invest. It can be a bit of an investment. You have to buy a separate ChIP reaction to go in with it, but it is important. And I would definitely seek to use a spiking control. It'll give you some real good feedback on what's going on. You have to try and be consistent the best you can with washing. With washing and not bead loss, okay? It can be, there are ways to minimize bead loss, okay, with a magnetic or if you've got a column, but if you start losing stuff on the way, you will almost certainly affect your results at the end, because the sensitivity of the stuff at the end stage, there's a lot of sensitivity there. That means there can be a lot of room for movement. So you take your ChIP reaction, your antibodies in there, it's gone overnight, and now you are going to capture your antibody, which is now obviously gone for your target, whatever that is, transcription factor or histone modification, whatever that is. So you, in the lab, you may already have tools to do this. People may already do IPs, they may use magnetic beads. So these are, the capture methods usually protein A or protein G. Make sure you have the right protein A or protein G for your antibody, okay? Because they don't universally go for the same species. Magnetic beads, things like daunting beads, they've been used for a long period of time. Some people favor agarose. There's a difference in the capacity of these things. Recently, I have stumbled across something called Recent Biosciences, and they sell a bead that has a really high capacity, and they're also hypermagnetic. So the kind of in the tube, they bind to the magnet very quickly, and you can put less beads in, which ultimately gives you less background. That's what I look at this. Agarose beads have a very little background, I often think, but if it's not in a column, you haven't got a column system, you've got more chance of getting bead loss, I think. That's maybe my opinion. But I've favored Recent Biosciences and their high capacity. They're kind of a porous bead. These actually, it's slightly different. It's a porous bead, and they do bind a heck of a lot of IgG, basically. There's a lot of protein protein G on there. But if I was starting off in the beginning, I'd just get agarose beads. Protein G covers most bases, that's protein G. Agarose beads, good place to start. Easy to see in the tube. It's a short stage anyway. But again, there's a chance for protein for bead loss in these processes. So the elution conditions. Now I've got some data at the end of this to show you about elution conditions. So you've now bound your complex to your antibody, and it's bound to your bead. And now you're going to wash all the way everything else off, and you've just got your complex together, and then you're going to elute off. And this, I think, is kind of an underappreciated step in ChIP, because it would be easy just to stick those beads with loads of SDS and even proteinase K, and shove them into a heat block at 65 degrees overnight, and you will literally strip everything off, and it will all come off, and you get loads of DNA. But the upshot of that is you've also got the background DNA as well. This is basically a challenge between the specific stuff and the non-specific stuff. And the more non-specific stuff you have in there, it will affect your fold enrichment at the end. And it's much better to go for a more specific elution, although you're going to get less back. You're going to get less material back. If you're doing like ChIP-seq and you pull them all together, you're going to get less. You can get a lot more if you just literally heat it up. But then most of that is the junk DNA, to use that old term. So we don't want that. And this has been shown actually for things like for doing a mass spec co-IP, the sister approach to this. It's much better to have a very, very targeted elution. So some low levels of SDS, just to break the antibody target bond, so you get less of your IgG in the fraction as well. We don't care about that with ChIP because we're going to get rid of it. It's going to be gone anyway. But the DNA has the same kind of, it sticks to the resins and things anyway. So then we are basically, we need to increase, we want to get lower background and only our specific DNA. So kits that have specific elutions are a lot better than I think the ones that just say, shove it in. The trouble with this is that in this protocol, in a lab, people will get their own opinion about this, and they will do it they may change bits here and there because it didn't work so well. There's very little DNA back there. So they then increase this step. We'll just put it at the 65 degrees overnight approach. I feel as if you're doing something that is that you're looking for a subtle change in binding, you need a specific elution for that. If you're just seeing whether it's yes or no there, maybe it doesn't matter so much. If you're just looking for a yes, no, you're just comparing it to virtually nothing, then that perhaps maybe doesn't matter so much. But I think that definitely specific elution, so look for that. Okay, now we've almost changed techniques now. We've now gone from cell culture, protein stuff. Now we're at DNA quantification. Now we've become a different scientist, like labs that only do DNA work. Now we're in a different realm, okay? And this makes it, that's why I think this technique is challenging. So we're going to quantify the DNA at the end. So if you're doing a regular ChIP experiment with this like a plate of cells and you're looking for something, you've got a small amount of material, don't try and measure the DNA at this stage with any other way than qPCR. So don't go and try and nanodrop your sample at this stage, all right? Because you're not going to get it back. There's nothing that you can actually detect. It's way below that level. So I favor qPCR at this stage. I don't do gels. I actually favor digital PCR, and I'll show you that in a minute, but that's not such a commonly found machine. Most people have access to a qPCR machine. And the way I quantify ChIP, now when I first started doing ChIP, I didn't do it this way actually, because I just really didn't know any better, I decided. But the best way of quantifying your ChIP DNA is to use a standard curve. And for every single primer you have, use a standard curve. Okay, so for every priming set you use, take a standard curve. Now you need 2 primer sets to quantify your ChIP DNA. You need a test, take the test where you're going for, gap DH, whatever it is, the gap DH promoter, something like that's good to start off with. And then you need to have a negative region. And you can buy these regions in. So it's a region that doesn't bind anything. It's like some desert island of nothingness that's out there. And that's what you need. You need to compare a test region with a negative control region. Okay, that is quite important. I would avoid any other calculation methods. Definitely not fold over IgG. You see that quite a bit. Definitely not percentage input either. I don't like that either anymore. I would definitely go for fold over. So for each ChIP reaction you do, you have to do 2 qPCRs, one on your actual region, one on the test region. Then you're comparing like for like. Okay, you're not comparing apples and pears. You're going like for like. You've got your target, and you're calculating the amount of junk DNA in there, the amount of good DNA in there, and you work that out as a fold enrichment. So it's test region over negative region. That's the way I like to do it. I think that's the best way of doing it. Here below, I've actually got some quite cool sort of ChIP results with tissue. Well, that's a qubit result, so that does work because it was a very rich source of those modifications. And then this is a test on the gap DH promoter. So you can see the fold enrichment in the IgG and the fold enrichment in the acetylation mark and the fold enrichment in a methylation mark, this being an active, a marker of active transcription, and this being a marker of closed off chromatin. And you can see the massive fold enrichment with the, obviously it's gap DH in its own piece of tissue. So that's very likely. Of course, I know I'm not trying to show you that there's no take-home message there at all, but just showing you that I think this is a great way of doing it. You've taken into account primary efficiency, you've taken into account the amount you put in, because that was equal at the start, and you can calculate the input sample as well, and I'll show you that in a second. Okay, so this is now a step forward now. So done some qPCR. Now what about the next stage of PCR? So in this case, I'm going to show you some results with digital PCR and why I like it. So if you have access to a digital PCR machine, and it's becoming more popular, I would almost certainly put the effort into using it. You haven't got to make any changes to your primers. Most of them work quite well, but bulk fluorescence. So when we do qPCR, we have bulk fluorescence. We're just measuring fluorescence straight out of that well in that machine. Digital PCR seeks to put the reactants into small droplets. They're in oil droplets, basically. And then you have PCR reactions in a droplet. Then you bake those, and then you do the PCR reaction. Then you count the number of positive droplets versus negative droplets. And what that does is that gives you an additional orders of magnitude difference. Now, when qPCR, at that kind of high cycle time, when it gets in the 30s, becomes extremely variable, okay, really variable. And it's not uncommon for a ChIP reaction to live in that part of the PCR curve. Quite common, okay? So often we're dealing with like low expressed proteins, maybe transcription factors. This can be a challenge. We're not dealing with always dealing with histones. We're dealing with things that there isn't very much there. Because it lives in that region, that can make it extremely variable, and I've seen this myself a lot. So we switched to digital PCR because it gave us that a bit more reliability at the lower end. And quite often, things like the IgG control were truly negative. Whereas on a qPCR, you can get an amplification of water, can't you? You can see something at the bottom really easily, and that is nothing there, but you can get a signal. And digital PCR showed us that. So if you can have that machine, it is definitely worth making the investment into using it. Here's an example, okay? So here we've got our positive and our positive droplets in blue and our empty droplets in dark gray here. This is the input sample for a gap DH promoter or a negative region, okay? Some region where there's usually nothing binds to. Here we've got our IgG fraction. And if you look on the right over here, we've got our gap DH promoter with acetylation ChIP and our gap promoter with a methylation ChIP. And you can see there's lots here and very little there. Just visually, you can see, we then turn this into copies, okay? It becomes copies per microliter, and then we can actually quantify how much we have there. So I think it's great. And this, to me, at the time, was a bit of a revelation, because I spent a lot of time doing ChIP with transcription factors, and sometimes you're just such, there's so little there to make, it's difficult to do 24 samples of ChIP in one go. And if you're trying to do a proper experiment and look at knockdowns, it can be tricky. So you have to kind of scale it back a bit sometimes. And this gave us a chance to be able to keep some scale in and also up the sensitivity. And it was a revelation at the time. It also can highlight something like quite nice things as well. So this particular example here, where we've got a sonication pattern. So I would always go for a tapetation sonication. I didn't mention it earlier on so much, but if you want to check your sonication, the DNA gel sometimes can be one of those problematical things to do where it can look very different from day to day. I've had that a lot in the past. But if you do a tapetation, I think actually it looks a lot better. And you can kind of see what's going on a bit easier as well, because you always see the DNA. So 2 examples here where we've got a difference in sonication pattern. And then when we run our results, so here we go on here, we've got each of the samples there, one for a target gene, which is TAT in this case, don't worry about what that is, but just a target gene. This is the negative control, and here is the spiking. So we put the spiking chromatin in the sample, we add a spiking antibody for a histone, and then we have a positive control for the spiking as well. So it's a whole separate ChIP reaction just in that sample. And you see the spiking is quite similar, but the difference here is the negative control DNA is much higher. And that's affecting, obviously, the sonication pattern isn't so favorable. Sorry, I'm going to move this screen out of the way a second, sorry. And the sonication pattern here isn't quite so favorable. It could be longer, heavier amounts of DNA there, and that's given us a higher background there. Another good example here is the elution conditions. Now look at this. This is interesting, I think. So we have 2 elution conditions. Okay, so one is like, that should say B there. Apologies. I'll change that in a second. I should say B on there, but A and B. A is the specific elution buffer, and B is the overnight heat method, shall we say, with SDS. And if you look at the amount of negative control DNA that's in between A and B, it's much, much higher. So yes, you've got more of what you wanted, but you've got a heck of a lot more what you didn't want. And also the same for the spiking too, it affected that as well. And actually it changes the fold enrichment. It's now a lot lower because you've now got a load more of this negative control DNA. Okay, and very finally, about the spiking control. So this using the spiking control illustrated to us how variable ChIP can be. And if you look across, say, 40, 50 odd samples of ChIP samples, you can see that there's actually a huge amount of variation. And having this in there can really help control this variation. And just very quickly, finally, if you are going to upscale the ChIP sequencing, remember that you have to combine lots of smaller reactions to get this to work properly. You can't just do one huge ChIP reaction, that doesn't work. You generally only get between 5 and 10 nanograms anyway, but I would take lots of smaller reactions, times that by 5, at least 5, and see how you go from there. You combine it before the purification stage. You never try and combine it afterwards, otherwise you're just going to lose all your DNA. Never take purified DNA, add it to a column. That is a disaster. You're all going to end up with tears. Okay, anyway, and just very quickly, a summary. So ChIP is a challenging technique. It takes at least a week to do it. If you're going to start off with some really easy materials, and I would always recommend a kit for ChIP sequencing because you can be sure about cleanliness and you can ensure that you've got reagents that are truly molecular biology grade. Okay, thank you very much.
Hi, sorry. Thank you so much for that really great talk, Toryn. If you could just go to the next slide, please, and I'll just quickly go through So just briefly want to touch on the protein tech antibodies that are available for ChIP. So all our antibodies are made in-house and undergo extensive QC by our really experienced R&D scientists. We actually have over 3,000 targets which have been validated by knockdown knockout, which is an incredibly important aspect of ChIP, that you have a antibody that's specific for your target. And all our polyclonal antibodies are made against full-length immunogens, meaning that they are really flexible and available to be used in many different applications. And over 4,000 of our antibodies have actually been validated or published in IP. And as Toryn said at the start, if you don't have an antibody that's necessarily specific for ChIP, as long as it's been validated or published in IP, then you can be reasonably confident to start there. We actually have around 220 antibodies published by our customers in ChIP already. So yeah, there's a really good starting point with the protein tech products for your ChIP experiments. Next slide, please, Toryn. And as I said at the start, we acquired the company Chromatin in 2020, and Chromatin are an antibody company. So essentially, what is an antibody and how can they be used for ChIP? So an antibody is the binding domain of a camelid heavy chain antibody. It's actually really small. It's only 15 kilodaltons versus the 150 kilodaltons of a conventional antibody. Nanotraps are nanobodies which are conjugated to beads, such as agarose or magnetic agarose. And the benefits for them in ChIP and IP is because they are fast and reliable and have one step. They have incredibly low background and non-specific interactions, purely because they don't have any protein A or G involved, meaning that they therefore have this much cleaner background, as Toryn was mentioning earlier. They also have an incredibly high affinity to their protein, and so they're able to precipitate really low protein amounts. And these nanotraps have over 60 publications for ChIP, and they are available for tags such as GFP, mCherry, and FLAG. Next slide, please.
Sorry, come in.
No worries.
There we go. Sorry.
And yeah, I just wanted to let you know that everyone who's attended today or is watching this recording is able to claim a free sample of any of our antibodies or a free nanotrap sample as well. We'll send these details out in your follow-up email. And then next slide, please, Toryn. Just to say thank you so much for attending, and Toryn will now answer any questions. Mike from Active Motif has been really great in actually answering a lot of the questions in the chat. Toryn, you've got the questions as well, because there's so many. I don't know if there's any that you think are particularly great that you could answer. There's a lot of thanks. Thanks, everyone. Really, yeah, Toryn, that was a really great question.
I'm trying to think what's the best Q&A ones. I go for those. So testing, sonic testing. So one question asked, how do I test if a double fixation is working? So this is kind of, and also it's about looking at a control protein as well. So so using DSG and these other kind of cross-linkers, it has more history in mass spec actually. I think it does anyway, because cross-linking mass spec has become, really interesting technique. I started off just looking at the DSG type fixes, and seeing whether on a Western blot I could see some high molecular weight bands forming on a Western blot. That's quite a good place to start actually, because you take the protein you're interested in, and then see whether you add with DSG, and then do a blot, and you should be able to see whether you get higher molecular weight stuff. Now, it doesn't happen so readily with formaldehyde, but it does with things like DSG. So just have a look and see. Control proteins, actually any protein that has a good, so you need a clean, you need something that's very, very clean. So you could do a histone antibodies are great, and you could try that maybe, or anything that obviously isn't huge size, it would be good as well. So something around sort of 60 kilodaltons, even for that tubulin maybe. I tried this with the glucocorticoid receptor before. That worked quite well. But if you have a good clean antibody, add your DSG and see whether you can get some high molecular weight stuff. Now, if it's not been published for your transcription factor, that's something else, of course. And you may just have to run sequential ChIPs and see whether it looks like it works. But it's difficult these things are tricky these are difficult things to optimize. But trying a Western blot festival wouldn't be a bad idea.
I've written a couple of questions down here, if I can.
Oh yeah, sure, yes, yeah.
Good. One of them says, you said that you have done a lot of ChIPs for transcription factors. I read that you should fix the cells longer than for histones. What do you recommend?
I don't know whether I've ever seen any. Mike may have some additional different opinions on this, but there's a danger to overfix if you're not careful. And sometimes for some, I think cell types, actually overfixing is definitely a bad thing. I don't know whether you should actually change the fixation length depending upon the target itself.
We agree, Toryn. we don't choose the fixation time based on the chip target, but rather the cell or tissue type. And we've dealt with so many different cell types that we have a different fixation protocol that we perform for different cell types. So we tell our customers to start with 10 minutes and 1% formaldehyde. And if that doesn't work for you, then try a little bit longer and a little bit shorter fixation times. So every variable needs to be optimized with ChIP, and that includes fixation time, sonication, antibody selection, chromatin prep. If these are many different variables that need to be optimized, and you will have to continually optimize them as you switch to different cell types and different antibodies.
Yeah, that is a great point. It is quite a process to suddenly change something is difficult, and also for tissue as well, actually, because tissue can be how quickly you can chop it up in tiny little bits as well. That's the other thing. If you're doing cell line, you just got to add it up. But with tissue, it can be variable from person to person. Well, I've seen before where people have been very good at slicing up really quickly, and then you're making the small pieces so that it gets in. Whereas if you've got bigger lumps there, it just is purely a diffusion process, isn't it? In this respect.
And speaking about sample prep, Toryn, there's a question from Rebecca Sizer who says, can the confluency of cells at the start of the experiment affect the results? For example, 100% confluent cells could have different protein binding patterns to 80% confluent.
I guess that depends upon your question you're looking at, I assume, in some respects. I mean, you probably not best use cells like that anyway to get really meaningful results. I mean, if you're adding a stimulus, of course, as well, then that can be, depends what you were doing, because sometimes you may have them complement like that if you were kind of, synchronizing them in a kind of circadian sense. But yeah, there could well be, a difference with that. I don't know whether it affects actually the kind of sonication of things, if it makes it any harder, if depending upon the transcriptional state of the cell, possibly. That, I don't know, that I could be straying into sort of like voodoo there a little bit. But I mean, certainly your results in the end could well be affected by the confluency of the cells, further down the line. But looking at some of the targets you're looking at, for sure, yeah, I would always be a bit careful of that. You're probably better off not ever having them at that confluency unless you've got some specific reason to do so.
And another protocol question is from Kriti, and she's asking, what should the optimum amount or sample to bead ratio be? And does adding more bead increase the binding efficiency?
I think it will affect the kind of binding efficiency as such. I mean, you generally, obviously, you're only putting in quite a small amount of beads into the sample itself, about 30 microliters of the sort of standard preps you get. It can affect the background, I always think. Adding more of the material into the chip itself will almost certainly could give you a higher background because you've got more stuff there combined what's in the sample. I favored the recent biosciences beads in recent years because of the fact that they have a very high capacity and we put a lot less in and it can bind the same amount of IgG out. So I'm always trying to minimize the amount of stuff going in because I always feel as if that is going to give me potentially a more of a background problem as time goes on. Remember that cell agarose in protein G is redesigned for antibody purification on a column, according to this idea where you run it through. So active T cell, the little columns, that's a good place to go for because then it's not actually, you're just running it, you're just capturing it, which is a really good idea, I think, actually. That's a good idea. And you've got a bit more control over the flow of things as well. But I think optimum ratio, I mean, for a chip reaction, I think it's just a question of you can only put so much in anyway, otherwise you'll have more beads than anything else. And usually around the sort of 30 to 50 microliters amount is okay. I mean, per reaction. And then in that, you want about 30 micrograms of chromatin. That's about as much as I've gone up to. I've tried higher ones before, like even up to the 50. And sometimes it's difficult to get samples that all have enough. Sometimes it's hard to get samples that have enough concentration so you can afford that at times. But If you have something like liver, of course, that's different. But I think keeping it to the smaller amounts is a much better idea.
And I think we have time maybe for one more question. And this is more of an analysis question, and it's from Reem, who asks, this is a more bioinformatics sensitive question, but do you recommend any specific programs for rescuing multi-mapped reads?
Multi-map reads.
Does that make sense?
Mike, have you got any
Choose a
ideas?
different question.
I don't. I'm less fay with the.
So I'm not also not a bioinformatics. I'm not a bioinformatics expert, but what we use we use Max2 for alignment and quantification, and I'm not sure if that program has a way to in your demultiplexing to get rid of the multi-mapped reads. So whoever asked that question, please connect with me afterwards, and I will get in touch with our informatics team and get you a better answer to this question rather than just guessing.
Yeah, I'd be guessing as well. I think
there's loads of really great questions that we haven't actually had time to answer, but we'll get a transcript of all these questions, and I'll forward these to Toryn and to Mike, and I'm sure they'll be able to get back to you within the next week or so in answer to your questions if we haven't answered them already. But yeah, I think we're over the hour now, and just to say huge thank you to Toryn. Thank you so much for your time. You've got some amazing comments in the chat section. Thanking you for such a great informative talk, and I really reiterate that it was really clear and really easy to follow. So thank you so much, Toryn, and thank you again to Mike for being on this call. Really appreciate all your help and for your help in the chat as well. That was really helpful. So yeah, just to say thanks to everyone, and we'll send a follow-up email, and I'll make sure that Toryn and Mike are able to answer any questions that you have.
That'd be great. No problem.
All right. Thanks. Bye.
Bye.